Corn leaf blight is the main corn leaf disease, also known as gloom mold. It is widely distributed and seriously harmful, stripe disease, leaf blight, and so on, mainly occurring in cold, cool, and humid areas. It was first reported in Italy in 1876 and spread all over the world in the early 20th century. Its occurrence was first recorded in 1,899 inches. At present, the occurrence covers an area of about 70 million mu per year, which is moderate in most areas of northeast, north and southwest China, and more prone to highly susceptible cultivars in some areas of northeast China. In a year of large-scale occurrence, the yield of corn is generally reduced by 15 to 20%, sometimes even greater than 50%. So, how to distinguish the corn leaf blight? Corn may be infected with leaf blight during its whole growth period. It mainly damages leaves, leaf sheaths, and bracts, among which leaves are the worst affected. Generally, it starts from the lower leaves of a plant and gradually expands upward. It rarely occurs in seedling stage and gradually worsens after tasseling. At the initial onset, small watery grayish-green spots appear on the leaves, and then gradually extend to both ends along the veins, forming spindly or fusiform large spots. The center of a spot is yellowish-brown or grayish-brown, and the edge is dark brown, and the spot is generally 5 to 10 centimeters long and 1 centimeter wide. At the final onset, multiple spots are connected into a large irregular wither spot. Those on some disease-resistant cultivars extend longitudinally and show brown necrotic stripes, causing the plants to wither early. When the field humidity is high, a lot of black mildew layers are generated on the spots, namely the conidiophores and conidia of the pathogen. Essentials of diagnosis: large fusiform spots appear on the leaves, and there are dark gray mold-like substances at the affected parts. The teleomorph pathogenic bacteria of corn leaf blight are Cedosphaeria turcica, falling into Cedosphaeria ascomycotina. The anamorph ones are Helminthosporium tursicum, falling into Axarohilum deuteromycotina. Since the middle and late 1980s, the physiological races of the pathogenic bacteria of corn leaf blight have undergone complex differentiation. Now we know that there are 16 physiological races, including 0, 1, 2, 3, N, 1N, and 2N, which means that the degree of variation in this disease is quite complex. The conidiophores are olivaceous, cylindrical, mostly solitary or 2 to 6 inches clusters protruding from the stomata, generally unbranched, upright or knee-shaped bending at the upper part, with 2 to 8 diaphragms. The conidia are straight or curved, grayish olivaceous, mostly with 4 to 7 membranes, thin at both ends, wide in the middle, fusiform, and grow attached to the top or curved parts of the conidiophores. The basal cells are tapered, and the apical ones are blunt round or oblong. The umbilical point is obvious and protrudes out of the basal cells. Germinal tubes are generated from both ends during germination, and chlamydospores are often formed during overwintering. For the leaf blight, the temperature of hyphae growth is 1035 degrees Celsius, and the pH is 2.6 to 10.9. The temperature of conidia formation is 1533 degrees Celsius, and the temperature of conidia germination is 5 to 42 degrees Celsius. After the spores are formed, they have a high ability to resist drying and can survive on corn seeds for more than 1 year. In addition to damaging corn, the Cedosphaeria turcica can also infect sorghum, Sudan grass, barnyard grass, and wild corn. The pathogenic bacteria of corn leaf blight mainly overwinter in the form of mycelia or conidia on affected corn residues in the field. Those on seeds and the affected residues that have not yet rotted in compost can also overwinter and become the primary source of infection in the next year. During overwintering, the conidia become chlamydospores due to the concentration of protoplasm and the thickening of cell wall. A conidium may form 2 to 3 chlamydospores, which have high stress resistance. After overwintering, the conidia germinate and produce invading hyphae, which invade directly through corn epidermis, and a few may also invade through stomata. Under humid conditions, the affected parts produce a large number of conidia, which are transmitted with wind and rain for reinfection, maybe multiple reinfections during the whole growing period of corn. Especially in blended zone of spring and summer corn, reinfection is more frequent because the former provides a lot of bacterial sources for the latter. Different corn cultivars have distinct disease resistance. If a susceptible cultivar is planted over a large area, it often leads to the prevalence of leaf blight. Generally, when the temperature is 20 to 25 degrees Celsius and the relative humidity is above 90%, it is conducive to the occurrence and spread of the disease. In the northern corn belts, the temperature from June to August is mostly suitable for the onset, so rainfall has become the determinant of the severity of leaf blight. Low temperature, rainy and humid days, and insufficient sunshine in July and August are all conducive to the occurrence and prevalence of leaf blight. Besides, after corn heading, it will be worse in case of insufficient nitrogen fertilizer, higher density, and divided by or poor drainage. No matter it is spring corn or summer corn, it would be worse in case of late sowing, dense planting, continuous cropping, closing to villages, and low-lying plots.